Welcome to Do You Know, where we dive deep into the wonders of science, history, and technology. Today, we're exploring something that connects every living thing on Earth: DNA. This microscopic molecule holds the key to life itself. So, let's unravel the history, structure, and incredible significance of DNA. Ready? Let's dive in. If you enjoy our content, make sure to like, subscribe, and hit the notification bell to stay updated with our latest videos. DNA stands for deoxyribonucleic acid. It's the molecule that contains the instructions an organism needs to grow, develop, survive, and reproduce. Think of DNA as the blueprint for building and operating a living being. From humans to bacteria to plants, DNA is the universal language of life. DNA is made up of molecules called nucleotides. Each nucleotide has three parts: a sugar molecule, a phosphate group, and a nitrogenous base. There are four types of bases: adenine A, thymine T, cytosine C, and guanine G. These bases pair specifically, A with T and C with G, forming the iconic double helix structure. It's the fundamental blueprint for all living organisms. Fascinating, right? The journey to understanding DNA began in the mid-19th century. In 1869, Swiss chemist Friedrich Miescher made a groundbreaking discovery that laid the foundation for modern genetics. Born into a family of physicians in Basel, Switzerland, Miescher initially trained as a medical doctor, following in his father's footsteps. However, his keen interest in research led him to the laboratory of Felix Happé-Seyler, a renowned physiologist and biochemist at the University of Tübingen in Germany. Miescher's assignment at Happé-Seyler's lab focused on studying the composition of white blood cells. These cells, critical for immune function, were difficult to obtain in large quantities, so Miescher sourced them from discarded surgical bandages soaked in pus, a surprisingly resourceful yet unpleasant method. Through meticulous work, Miescher isolated a novel, phosphorus-rich substance from the nuclei of the white blood cells. He named this substance nuclein, as it was found exclusively in the cell nucleus. This was a remarkable achievement because it was the first identification of what we now recognize as DNA. However, Miescher's journey wasn't without challenges. The laboratory techniques of the time were rudimentary, and isolating pure samples was an arduous task. Contaminants and the fragile nature of cellular material often led to failed experiments. Additionally, the significance of nuclein was not immediately apparent, even to Miescher himself. He initially believed it was involved in cell energy storage, similar to proteins, and he didn't realize its role in heredity. Despite these limitations, Miescher's discovery was a monumental step forward. Although the scientific community at the time didn't fully grasp the importance of nuclein, Miescher's work laid the groundwork for future breakthroughs. His findings would inspire other scientists to explore the mysteries of the cell nucleus, eventually leading to the identification of DNA as the carrier of genetic information. This was the first step toward discovering DNA. Fast forward to 1952, Rosalind Franklin, a brilliant scientist, played a pivotal role in uncovering DNA's helical structure. Born in London in 1920, Franklin came from a prominent Jewish family that valued education and community service. She excelled academically, earning a degree in physical chemistry from Newnham College, Cambridge. Despite facing significant barriers as a woman in a male-dominated field, Franklin's determination and intellect drove her to pursue groundbreaking research. Franklin's career led her to the Laboratoire Central des Services Chimiques de l'État in Paris, where she mastered X-ray crystallography, a powerful technique used to study molecular structures. In 1951, she joined King's College London, working in John Randall's laboratory. Here, she focused on understanding the structure of DNA. Using her expertise in X-ray diffraction, Franklin meticulously prepared DNA fibers and captured high-resolution images of their structure. One of her photographs, famously known as Photo 51, revealed the characteristic X-shaped pattern that suggested DNA's double helix. This was a monumental achievement, as it provided critical evidence of DNA's three-dimensional shape. But the breakthrough came in 1953, when James Watson and Francis Crick used Franklin's data to model the double helix structure of DNA. James Watson, an ambitious young American biologist, and Francis Crick, a British physicist with a keen interest in molecular biology, formed an unlikely yet dynamic partnership at the Cavendish Laboratory at the University of Cambridge. Watson, born in 1928 in Chicago, Illinois, had a strong passion for genetics and studied zoology at the University of Chicago. By the age of 23, he had earned his PhD in zoology and focused his research on understanding the structure of nucleic acids. His youth and ambition made him a bold thinker, eager to take on the mysteries of DNA. Francis Crick, 12 years Watson's senior, had a background in physics and initially worked on radar during World War II. After the war, he shifted to biology, driven by a fascination with the molecular processes of life. Crick's experience in theoretical problem-solving complemented Watson's biological perspective, making them a formidable team. Together, they approached the DNA puzzle using a combination of physical modeling and theoretical analysis. While they didn't conduct their own experimental work on DNA, they relied heavily on the data from Rosalind Franklin's X-ray diffraction images, Maurice Wilkins' research, and the earlier chemical findings of Erwin Chargaff. Chargaff's rules, showing that adenine pairs with thymine and cytosine with guanine, provided a critical clue to the structure. In February 1953, Watson and Crick constructed their now famous double helix model. This elegant structure demonstrated how DNA could replicate itself and carry genetic information through its complementary base pairing. Their discovery not only explained the molecule's function, but also revolutionized biology, marking the beginning of the modern era of genetics. They described it as a twisted ladder, revolutionizing our understanding of genetics. Watson and Crick won the Nobel Prize in 1962, but it's crucial to acknowledge Rosalind Franklin's immense contributions, which often go uncredited. Now, let's see DNA in action. DNA resides in the nucleus of cells and is organized into structures called chromosomes. Humans have 46 chromosomes, 23 from each parent. When cells divide, DNA replicates itself. The double helix unzips, and each strand serves as a template for a new strand. This process ensures that every new cell has an exact copy of the DNA. DNA also makes proteins, which are essential for life. Through transcription, DNA is converted into RNA. Then, during translation, RNA assembles amino acids to form proteins. These proteins perform countless tasks, from building muscles to fighting infections. Ever wonder why you look like your parents? DNA is the reason. Your DNA is a unique combination of your parents' DNA. It carries genes, the basic units of heredity, which determine traits like eye color, height, and even susceptibility to certain diseases. Modern DNA testing allows us to trace ancestry, identify genetic conditions, and even solve crimes. It's amazing how much information is packed into something so small. Today, DNA is at the heart of cutting-edge science. In medicine, genetic engineering and CRISPR technology are revolutionizing treatments for diseases like cancer and genetic disorders. In agriculture, DNA research has created genetically modified organisms, GMOs, that resist pests, grow faster, and adapt to changing climates. DNA is also used in forensic science, helping solve cold cases and identify unknown remains. While DNA research has immense benefits, it also raises ethical questions. Should we edit human embryos? What are the risks of creating genetically modified organisms? These debates challenge us to use DNA technology responsibly. Here are some mind-blowing facts about DNA. If you uncoiled all the DNA in your body, it would stretch to the sun and back over 600 times. Humans share 60% of their DNA with bananas. Yep, bananas. DNA evidence is so precise, it's used to identify suspects with a 99.9% accuracy rate. DNA is the foundation of life, the code that makes us who we are. From its discovery to its modern applications, it has changed the way we see ourselves in the world. And as we continue to unravel its secrets, the possibilities are endless. If you enjoyed this video, give it a thumbs up, share it, and don't forget to subscribe to Do You Know for more fascinating topics. See you next time.